Okay, let's start off the Monday's class. As always, if you can hear me, you can raise your hand or leave your chat. You can hear my voice. Okay, so it's working. So, okay, so we are start off discussing the format of the sequencing data set we get from the next generation sequencing machine. And I mean, I put the title called data modality, and this will cover various formats of files you get from the sequencing data set. And the main idea of the course today and also next week, we are learning different, the formats of the files. So the format means that, you have to understand how we put the notation for different sequence information into the file, and also how we deal with those information into the research. So you have to make the connection between the sequencing information, DNA information, to the data file format. And I would say this is probably the very unique time to learn about the latest sequencing data set, and also you probably find quite awkward, because you never learned about the data format in your previous learning. So if you have any questions, it's always encouraged to leave you some of your questions onto the Slack channel. And also we are covering the data set of different process of the sequencing pipeline. So this is like the situation you learned about, what is the MS PowerPoints, the Word, or the whatever the HWP program. So this is, you feel probably like learning the new the concepts like computational things, but yeah, this is about that notion. Okay, so what we are going to learn today, we are talking about the workflow of genomic sequencing from the DNA to data. So obviously, we collect the DNA from someone, samples or patients. Then we extract the DNA, then we put it into the sequencing machine, then we generate the data set. So we are covering sort of the first part of those workflow. And then once we get the sequencing, then we get the data from the machine, and we call that data raw data, because it's from the the machine, and we have to process this raw data into certain format. Then we can process in a downstream analysis. Then we can find mutation, or we can estimate the expression, or whatever you want, right? So, and this is the part of the process we call the raw data and Fastq. And then once it's processed, then we specifically call it the alignment, right? So alignment for sequencing reads, we call the line reads. And then we specifically put it into certain format, which is SAM, BAM, or CRAM, right? And then during this alignment process, we use the strategy called hash mapping. Then we learned about the hash-based mapping, and this is based on the KMA, right? And all of this processed in the send file format. So this is another format we have to learn in the sequencing data set. So if you read this list of the the items we discussed today, this is totally new thing for you. So you never learned these things before. So please focus on the the lecture, and also come up with the questions if you're not clear about that. Okay, so the process of the generating the genome sequencing data. So as I mentioned, that if we collect the DNA samples from patients or mouse, or even sometimes if you do experiments with flies, fish, or frogs, then you get the DNA from the the biological organisms, right? Then once you get the DNA, then you make the library out of these DNA samples, then you put it into the sequencing library. Then once the DNA, DNA materials transform into the library format, then you can put this sequencing library into the machine, right? So as we learned in a previous time, it depends on the sequencing machine. So if you use the Illumina machine, then you have to make the library only for Illumina sequencing. And if you use the nanopore sequencing machine, then you have to preparing your library for the nanopore sequencing machine. So we can say you prepare library, or you can use the abbreviation. You prep the library, then put it into the machine, right? So I mean, I can guess that in your career you either work in a Illumina machine or PacBio or nanopore. So I normally say you do the library into the Illumina machine. Okay, and once you put your library into the Illumina machine, so I mean, put the library into the a machine means that library is liquid into the liquid format. So it's in the tiny tube. Then you use the pipette and move the library into the machine. Then the machine will start analyze those library information into the data format, right? It's a data format means that here data is the sequencing data. So there has to be the DNA sequence, right? So that information at the first time stored as a image format. So if you recall the previous topic, we talked about you use the laser to detect the fluorescent, the intense. So you basically transform those light information, fluorescent information into the image information, right? So the first format of the data set you get from the sequencing machine is the image, right? Then that image information is translated into the text, right? So this is the technology native in a sequencing machine. So transform the image information into the sequence information. Then the sequence information translate into the DNA information. So normally that the sequencing data set, the raw data set, looks like this. So that has the ID at the top. Then at the second line, it has the DNA sequence. And the third line, it has the separator. So separator means that it doesn't have any information. It's just the separating between lines. Then the last line, which is the fourth line, is the quality information, right? So all of these four lines are forming a chunk, single chunk, and that information represent the DNA sequence information you get from the certain area within the flow cell, right? And if you remember the situation when we do the Illumina sequencing, we normally do like a million reads or sometimes billions reads. So lots of reads from the sequencing machine. So that means that reads represent this information, right? So this information. So if you do the 150 million reads, that means that you collect the 150 million chunks from the sequencing data set, right? So this is the lots of data set you get from the machine. Okay, so then once you get the sequencing reads information, then you put those reads into the alignments. Then after that you find the mutation from the sequencing machine. So this is the sort of the typical workflow of the genome sequencing. Then put it into the analysis of finding genetic variants from the samples. Okay, so here is the list of the data format you get from the sequencing machine and also related to this process. So the first step, we get the raw data, and raw data stored in Fastq format. So the format name is Fastq, and that contains the aligned read sequence with the base qualities, right? So this Fastq information is the one we see in this slide, right? And then once you get the raw data, you put it into the lineages to the reference genome, then you get the aligned reads. So basically your read sequence is aligned to the reference genome. Then you have the location and you know the positions of the reads where to bounce to the reference genome. So in that case, we store that information into the SAM format, which is the sequence aligned map format, right? So shortly we call SAM format, and that obviously contains the aligned or unaligned reads, right? So if if you're successfully aligned to the reference genome, then we call the aligned reads. But if you have failed to alignments, then we call the unaligned reads, right? And this is the text format. So text format means that if you open up with, if you open this file with certain program, then you can read it, right? So text format, literally the information stored as a text, right? Or if the information is stored in a binary format, so binary means that only computer can read that information, then if the information stored in a binary format, then we call the BAM, right? So SAM format, SAM file can be stored as a BAM file. Then this BAM file means the binary alignment map. So see the differences between SAM and BAM, obviously S and B, right? And S means that the sequence, right? And this information is the text file, and the BAM file is the binary file. So we shortly call BAM file. So the issue is that why we store this information in a binary format. The reason is that if you store the information into the binary format, that means that the binary requires certain interpreter, right? So interpreter here means that computational program. So computational program can be the Chrome browser or whatever the programs you used in your computer, right? So if you use the window, then the window has its own program, right? And I used Mac, so Mac has its own program. So we developed the program to do some work, to do some the aims, right? So that situation, we specifically developed program for the for certain tasks, right? So if you store the sequence information into the binary format, then you can do specific task, right? So if you want to find copy number variant, or if you find certain position quickly, then in that case you have to use the binary format. And this can be any the purpose of task, right? So you can build your program in any the purpose, right? But the point is that why we develop the format into the binary, because we want to do something, right? And also another point is that why we use binary, because computer takes binary much more quicker way, right? So if you use the text information into the computer, or if you use the binary information into the computer takes binary information much quicker than text information. So that's why lots of programming, not just the the sequence information. If you think about the gaming, right? So if you store your game into the the file, save file, then if you want to open up the save file with Microsoft Word, it's not going to open up, right? You will see lots of different alien character, because that save file only can be read by certain the game platform, like Nintendo or Steam, right? So that case, it's again they use the binary information, they enhance the speed, enhance the efficiency of certain tasks, right? So that case, also the sequencing program, we use the binary format rather than the SAM file, because it's much quicker, it's much efficient, and also it's much specifically working on a certain task. And also binary information is much more compressed, right? So if you store your sequence information into the text format, that will require large amount of disk space, right? So if you think about the disk space is takes money, right? So you want to store in a much more handy and much more lighter way, so you can save the cost for the analysis and you can save the cost for the storage, right? So cost saving is the critical part of the industry and also research as well. So you want to put the binary information for the sequence information. And even if you store binary information for your sequencing data set, it's not enough. Not enough means that, these days we get, petabyte or zettabyte scales of the sequencing data set. So, if you store those information into the certain data, the warehouse or data storage, it takes lots of money, right? So people come up with sort of nice development of how we compress more and more. So we basically use the advanced compression model. In that case, we use the CRAN format. So this is much recent way to store the information into the into certain format. So people developed the same format at the first time, then they came up with the BAM, and lately people use the CRAN. So if you analyze the whole genome sequencing data set and conventionally used in the research, and if you generate the file from one person, right? So if you generate the data set from one person, it will take 1 terabyte, right? So 1 terabyte is, probably larger than most of people's, computational disk space, right? So most laptops use 256 gigabytes or 520 gigabytes, right? So 1 terabyte it's amazingly large data size, right? So if you store that information into the BAM format, then it will take 200 gigabytes, right? It's still big, right? So if you sequence not just one person, if you sequence 100 people, it will take up like a 20 terabytes, right? So it's quite large. And then if you store that information into the CRAN format, it takes only 30 gigabytes. So it's almost 30 times less than standard format. Okay, so this is the second step data format. And the last format is the VCF, right? So VCF is the variant code format. And this is including the variant information, right? So once you get the aligned reads, then all of your DNA sequence information mapped to the reference genome. So from those information, you can estimate which position has the mutation from those read information, right? So this variant information contains variants and their related information like quality, depth, or position, or blah, 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 right? And then this variant, including, as we learned in a previous session, it's the SNP in their structural variation. And also this information also containing the sample information. So not just the variant position, it has the where the sample is from and which the samples belong to which pedigree information, right? And also variance is related to the consequence, right? So all of these variance consequence is also written in this VCF file. So we put the genome annotation for variance. So if you find one variance, then let's say whether this variant is missense or which genes or which transcript. So if you want to learn about the next generation sequencing, and you have to understand this NGS data format, this is sort of the 101 things for latest genomic research, not just for the genetics. I mean, I would say, almost, all of the students in this class will use the NGS technology in your future career, whatever you study, biology, biological medicine, or the biomedical research, or microbiology, or even the clinicians. We are living in the era of the NGS technology, and this technology is not really the exceptional things anymore. This is the daily data set. So the starting point is learning about the NGS technology is knowing the data format, right? So I mean, I got some students for the the whatever the time of whatever discussing their career or the consultant, and I mean people quite I mean come up with the sort of the questions about how we continue their career into the bioinformatics or genomics or whatever the sort of the data science things in biology, and some of I mean there are some demanding for this field, and because of this reason some of the course running on for the the biological data science or whatever the genomic course in in online, even from the the Korean government, but things you can learn from the class, because the knowing about the data format is actually linking the idea between the genetics and technology, and also the genetics and genomics. So nothing really the particular notion you have to go somewhere to study or for the extra hours. I mean knowing about the data format and think about what is the notation and format means in the context of the genetics and also the polygenic model, then I mean this is great starting point. You can learn about the sequencing technology in your career. So we are keep it in that notion and discuss how we understand the basic concept of the data format in an NGS format. Okay, so the first one is the raw data and Fastq. So raw data stored in a Fastq format, and this is a standard format for storing and defining sequences from the NGS technologies, right? So Fastq is a simple format for raw aligned reads, sequencing reads. And this Fastq format is a simple extension to FASTA format, which is the reference genome. So sequence and okay, so we have to look at the FASTA format at the first time. So if you go to Wikipedia of the FASTA, and the FASTA format is the file format storing the reference genome sequence or reference protein sequence in early days, right? So if you look at the FASTA format, it looks like this, right? So this is the I don't know, this is the mixed up of everything. So if you look at here, this is probably the protein sequence, and this is also stored in the FASTA format. So if you look at closely, it actually has one line here and the second line here. So it has the separator, but all of the sequence information, protein sequence information stored in here. So if you look at here, so I mean, this is the two lines. So first one is the ID, and the second one is the sequence. So can you guess this is similar to the one we just saw before? So if you record the Fastq format we see in here, right? So that one has the two lines. So the first line is the ID, and the second line is the sequence, right? And it has the third and fourth line. So this is the different things compared to FASTA format. So FASTA format doesn't have these two lines, third and fourth line. So third line is the separator, so it doesn't really have any meaning. But fourth one has the a certain meaning. So this is the quality scales, right? So if you look at the FASTA format, it doesn't have the quality. It just have the ID and the sequence information, right? But Fastq is different. So Fastq has the quality information, right? So sequence and associated per base quality scores, right? So it has the ID here and sequence information here, separator, and the quality scores. And each symbols is corresponds to individual nucleotides onto the sequence lines, right? So Fastq has the quality information. So that has the FASTA with quality. So we shortly call Fastq, right? So why we care about the Fastq? So not why we not storing the information in the faster format, right? So that is the starting point. So if you create the reference genome, people store the information into the faster format. But if you generate the NGS data, you put it into the Fastq format. And NGS data care about the quality, right? So we put the quality information into the faster format. So why we care about the quality? The reason is that the quality is not 100% highest quality, right? So that's why we put the quality information into the NGS sequencing information. The NGS or any measurement of certain individual experiments is not 100% perfect, right? So it's not always coming up with errors, right? So I mean, this could be quite, you know, awkwardly coming to you because, you know, when you do the experiment, if you use the great technology from the, you know, latest days, and your expectation is that machine has to be, you know, perfect, right? But always your measurement has some error, right? And if you're knowing about the, you know, the machine can have error, then that will change your perspective because you're always coming up with some, you know, the variance, and it always coming up with some deviation from your measurement, right? So you have to get some confidence, you have to build some trust from your measurement, right? So in that case, the statistics is always the best way to represent that confidence of your activity, right? So if you use the NGS machine, if you doing some activities called genotyping, then you have to get the confidence from your experiments. So in that case, we put the quality information into your measurement. So here we use the quality information of your sequencing process. So we put the quality scores into the sequence, right? And this sequence information, this quality score information stored as a ASCII2 characters, right? So we're not storing this information into the numeric information. The reason is that, you know, we use the numeric scales of the Fred scores, right? So Fred scores is the scales between 0 to 40, right? And it representing the number of errors you can get from the sequencing machine, right? So if you put the, let's say if you put the 40 in your first sequence information in here, right? And 40 is the 4 plus 0, right? 40. So it has the two character. So two character doesn't really match into the single nucleotide. So there is no way you can, easily represent those quality information into the single nucleotides, right? So people has to come up with different ways to represent those quality information. Then we use the symbols of the single character and matching to the single nucleotide, right? So in that case, we use the ASCII2 notation, and this is the one way people conventionally use for the computational way. So people use the ASCII2 character into the quality information. And of course, you don't have to memorize all of these ASCII2 symbols for the certain scores, but you have to understand we have to use the ASCII character for the single nucleotide representation, right? Okay, so that is the basic notion of the Fastq information. And also another things you have to understand is the ID, right? So the first line of the Fastq is the sequence ID. So here, so we're not gonna put very simple, simplified ID version like seq ID. Instead, we use more specific way for the sequence ID, right? So ID is identifier, I shortly call ID, right? So the first line of the Fastq format sequence ID containing the lots of information related to the flow set, related to the machine, related to your experiment, right? Okay, so it contains several lines of several items, and is delimited by colon, right? So delimited is another important terminology commonly used in computational field and also bioinformatics. So delimited means that the separator, but the delimited is more like used in a row wise. So separator, you can put it into the into the lines, but the delimited is the used between the items, right? So delimited is the separating the mark between the items here. So it has the how many items here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, right? So it has 11 items. So first item is the unique instrument name. So I mean, which machine you used in the in this experiment, right? So it has to be certain unique name, right? So not just the Illumina machine or Illumina HiSec something. It has to have certain name for the machine. So then you can easily tracking data set in future situation, right? So this is like, you know, the situation you see some ID in your, you know, if you buy milk or if you buy certain snack, then you can see sort of the manufacturing IDs of from the manufacturers, right? So this is the same situation as this, right? And then the second item is the run ID, right? So run ID is, you know, it literally which run you used in these flow cells, right? And the third ID item is the flow cell ID. So you're not gonna, I mean, you have to use the single flow cell for each machine, and this flow cell cannot be reused. It has to be one-time use, right? So you use the flow cell in one experiment, one run, then you discard the flow cell after that, right? So each flow cell has its own unique ID, right? And it has the manufacturing ID from the company as well. So all of this information is stored in here, right? And the next item is the flow cell lane, right? So if you look at the flow cell from the previous sections image, then it has the 8 or 10 lanes in a one flow cell. And if you inject your library into flow cell, you have to choose which lanes you're going to inject your library. So if you inject your library on the first, second, or third, or fourth, then it always recorded it into the sequence ID as well. Okay, and the tile numbers within the flow cell lane or XY coordinates from the tile, and this is all of the information about the physical position of the flow cell. So if you record the concept of the flow cell in a lecture, it has the tiny words in the flow cell and also the tile information, and all of this information stored in the computation, and that information is also stored in the Fastq format. Then you can easily track down, I mean, if there's any problem in a certain positions of the flow cell, then you can easily track down that area and you can discard that information from your analysis, right? Okay, let me give you a bit more TMI on flow cell. Illumina flow cell on here, right? Okay, so this is the flow cell, and we actually looked from here, right? So okay, so this is the Illumina flow cell, right? And here is the lane. So 1, 2, 3, 4, 5, 6, 7, 8 lanes here, right? And if you see the tiny holes, and you actually put the your library through these holes and put it into spread out across these tiles into the flow cells, right? And flow cells has the ID next to the hinge here. and also it has the barcode information from the manufacturing things, right? And then once you put your DNA libraries into here, then it will flow by liquid, it will flow by the pressure and spread out through the each tiles, right? So if you look at close, then it has the each tile information here, right? And each tile, if you look at individual tile, it has sort of these priming stations, right? And your library get into this each tile information and getting the priming binding and process the sequencing information. But why we care about this, the tile information and tile position, the reason is that back to here. So if you inject the library as a liquid format, then you spread out through the pressure, right? But sometimes the injection, their activities, it can introduce certain technical errors, right? So if you do the experiment, sometimes, you know, if you inject your library samples into here, sometimes air bubbles coming with the injection. So not just the liquid or not just the library, sometimes, you know, the air is coming into these flow cells as well. So what airs makes problem in this situation, air is coming through here, then it can block one of the tiles entirely here. And then those areas not gonna be sequenced, right? So there is no way we can get the good image measured from these areas, right? So we are caring about, you know, the tile position, and we are caring about, you know, which position has the error-prone information generated from the machine. Then we can, you know, we can dig out that errors and we can improve our experiments, right? So think about the situation. So this tiny flow set and these tiny experiments takes like $20,000. So if you do the sequencing in, you know, if you are hired in a sequencing company and if you conduct the sequencing experiments, right? And these tiny things takes like, you know, $20,000 and it, you know, it's a huge cost, right? So if you do the sequencing and if you get the research, that's good. But if your experiment has some problem, or if your machine has some problem, and if you get constant errors or the bubbles on this position, so then that situation you have to ask that, you know, is this my problem? Is this my sample preparation problem? Or is this the problem inherently from the machine or certain technical errors, technical defects from the, you know, library preparation process, right? So this is the things you have to keep the all of the information and you know, finding out sort of the errors from your failures, right? So all of the information has to be stored in a certain position, and the Fastq format is the one way you can do for that purpose, and that's why we're keeping that information for the sequence experiment as well. Okay, so that is the old information, the sequencing, the paired information here as well, and also when you inject the library into the flow cell lane, you're not gonna inject only one sample. You sometimes inject more than one sample. Sometimes you inject, you know, 4 samples, 8 samples, or 100 samples. So in that case, you attach the index sequence to your library. So this is like a unique ID for the samples, right? And if you, let's say, if you put 2 samples into the same flow cell lane, then you use the 2 sequences of the index. So one is the ATC ACG, and the other one is the TTA TTG something. And these 2, you know, 6-digit, you know, 6 nucleotide sequences totally different between these 2 samples. Then you can easily, you know, separate out from your in your process. Okay, so the first thing is the converted image from the Fastq information, that process we call the base calling, right? So getting the raw signal information into the raw sequence data set, which is the Fastq format. Okay, so I think this is the one we talked yet last time, the minus log 10 times 10 scales for the error rates, and this is we call the Fred scale scores. And this is the the log scales as I discussed before. And the Fred scale is greater to 20 is it's okay because it's 1 in 100 error rate. And also we talked about how the sequencing quality change over the cycle information. Okay, so and once we get the sequence information from the machine, then we do the alignment, because all of the sequence information from the machine is totally fragmented, and we don't know where where it's from, right? So this sequence information is randomly fragmented, so there is no way we can do chromosome 1's disposition or chromosome 2 disposition, right? So this is all randomly fragmented, and we have to build into the original DNA sequence order, right? So this process we call the sequence alignment, right? But this sequence alignment has lots of problem and challenge as well. The reason is the human genome is so big, right? So it's about 3 billion base pairs. But if you think about the Illumina machine, it only gives you 100 base pair or 200 base pair. So how we build up all of these things to 3 billion base pair scale? So that's one issue. Another issue is the sequencer can produce 1 billion reads per run, right? So lots of reads fragments from the machine. And also, if you think about the quality, the genome sequencers can make mistakes frequently, right? 1 in 100 is quite high rate, right? And accurate alignment takes time because it has to be with the computation, not by your hands, right? And it takes the memory and it takes CPUs. So, let's say if I do one person's genome for the alignments, you have to use like a 16-core CPU. So you have to use 16 cores. And also you have to use like a 24 gigabytes memory, right? So this is one person's genome. So think about the situation, you have to use, 10,000 people. You have to use like, 100,000 CPUs, and also you have to like more than 10 terabytes memory scale. So it actually does exist in a computational field. And if you use the Amazon Cloud or Google Cloud, then you can, use those resources for your sequence data set. So, for instance, my laboratory here at Korea University, we use we have more than 500 cores for the CPUs, and also about 2 terabytes memory for the process. And also when I used to work in California University, we use 10,000 CPUs and like a few hundred terabytes memory for all of the data sets. So computational things, it's always limited situation, but it can be possible, but always keep in mind that it takes some memories and CPUs. So the alignment is not always an easy issue to figure out, right? And also alignment strategy always, quite complicated. And also, the library preparation is quite varied by different technology, and the animals of your experiment. So you have to consider all of these experimental design for your alignment process. Okay, so alignment is the one of the central things to most genomic research, especially in early days. Alignment is everything about bioinformatics, right? So, getting the alignments in a quick way, in the more handy computational, framework. So if you do the alignments 100 CPUs and 2 weeks for the for one sample, then you come up with better algorithm of the alignment, and you only use 2 CPUs and done it in 2 hours, then you will be millionaires, because alignment is something everyone wants to figure out better and better situation, right? So alignment get the transform the raw data set into the certain format, then you can find the mutation out of that, right? So alignment is the the core part of the bioinformatics in data processing. Okay, and another problem is that, I mean, if you look at our genome, we are, half of our genome consists of the repeat sequence, right? So if you look at our genomic information of our data set, we have so many, the chunks of the repeated information in our DNA sequence. And those DNA sequence always gives you sort of tricky situation for the accurate alignment, right? So if you think about the jigsaw puzzle, and if you get nice pictures, this is the entire DNA sequence we are expecting from the one person's human genome. But if you fragment it into the sequence rate, like 100 base pair, 200 base pair, and these jigsaw puzzles, if you build this entire complete pictures, it takes some time, right? It takes some labors, it takes some, the hands for that, right? So best scenario is if you do the sequencing, the best scenario is you read the sequencing information without the limitation. So not just 100 base pair, not just 300 base pair. You can read 3 billion base pair in a one shot and no errors, right? So this is the best scenario. If you develop the technology, you would be billionaire. But unfortunately, the current technology, you can only read 100 or 200 base pairs, and then sometimes this read has the error information, right? So if you put the one DNA sequence like here, and one of the positions here has the T, this is the error, right? So this is not the nucleotide from the nature, right? So this is not existing in that person's, in that samples, right? But then the problem is if your data cannot, tell you at the first time that whether this information is the mutation or error, right? So think about the situation when you do Googling, right? I mean, if you are good enough for your finger, you have very sharp finger, very well operating finger, then you can type, correctly, right? But my case, I'm very always in a rush. So I type, most of the time, my typing was wrong. And the Google gave me, did you mean this, right? So did you actually meaning these words? And I can click the one. But in a sequencing alignment, there is no way we can do that, right? So if you get the error sequence from the machine, there is no way we can fix the error, right? So you have to keep in mind that in reality, it has lots of errors from the sequencing machine, right? So there's two ways we normally code for this process. So first one is the assembly, and second is the alignment. So we mainly talked about alignment. So what is the differences between these two? So assembly is the situation you build all of these read information into the entire original sequence information without reference genome, right? But alignment, you actually bring those reads binding to the reference genome. So the two differences between these two is the existence of the reference genome or not, right? So think about back to the jigsaw puzzle. So if you get these puzzles from your friends, and your friends said, complete this puzzle for the pictures, right? And she or he give you a picture. Then that picture is the reference genome. Then you know about the picture, then you following the pictures, then you can make the entire puzzles, right? But without this picture, there is no way. There is almost, almost impossible way to figure out the original pictures, right? So these two things are quite different. But then the question is why we want to use assembly, right? Why not we just doing the alignment? Why we have to do assembly? The situation is that, I mean, if you are working human, then we know that reference, if you're working mouse says reference, right? But if you are working in a certain species, not many people did genome research or no one did genome research ever, right? Then in that case, we don't have reference genome, right? So if you find the new species from the mountain, right? And let's say you find certain mushroom and never, no one never ever, looked at that organism in a genomic research, then in that case, you have to extract the DNA, then you have to assemble that information into the reference genome. That's that case, you have to use assembly, right? This is the same situation for COVID-19, right? So coronavirus 19, we don't have the reference genome at the first time. So you have to make the reference at the first stage. Then once you finish that de novo assembly, the reference genome, then you screen the lots of patients and extract the virus from the patients. Then you do the alignments, then you can guess whether the patient has the mutation or not, right? So you can predict which strain belong to, which strain of this person's virus belong to, which area, right? So the question is that, if you look at the newspapers or the media, then you said, this is the COVID-19 UK strain, or this is from the India. So new strain outbreak from somewhere by the, the stupid gathering. Then that case, how we can know about the information? Because we know the reference genome, then we can identify we can do the quick alignment and compare those DNA sequence information. So that's why assembly and alignment is both important, but for the situation, knowing the reference genome, then we can go with the alignment, right? Okay, that's good. And one quick TMI and advice. Working in an assembly situation or working the algorithm development is quite, quite hard and also quite rare and also very computational intensive work. But, flip side, or not flip side, but positive side is that if you really interested in assembly situation or developing the algorithm, then, not many people in the world can do these types of research. So, it's definitely quite worth. And also, and also another notion is that if you work in a, I want to do some ecology where I want to do some, I want to discover the new strains, then assembly is yours, right? So in that case, you can go into some of the computational biology books, especially for the algorithm part, and that will, that will give you sort of the intriguing sort of aspects for the assembly and also just genomic research. But the most of the topics we dealt in this class is the different notion. We are more onto the genetics part and also covering the how we understand the variant from the person or the population. So we are more onto the alignment situation. Okay, so the next topic is how algorithm is working. So the famous one is we are working on the hash-based mapping and also based on the k-mer, right? So hash-based mapping, so let's think about the situation. We had the toy genome like this, so it has the 16 base pair, right? And you basically create sort of the index of this genomic information. So let's imagine that we're not talking about the 3 billion genome, we're talking about the 16 base pair genome because we want to make very simplified examples, right? And you can basically build up the index. So index, how you understand, you can think it as a dictionary, right? So you can basically build up the list of the items from the DNA sequence, and also think about the all possible combinations of the DNA sequence. So one possibility is CAT, and the second one is ATG, TTG, GGT, blah, 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 and all of these lists, right? And then what can you do is you can measure, you can identify which positions these items from. So CAT is from the first positions, right? And ATG from the second position. And the third position, the TGG is from the third position, right? So you make all of this information into these types of the index, right? So CAT from the first position and the seventh position, and the TGG is from the third position and the 10th position. So why are we creating those index information for your mapping? The reason is that we have very long sequence of DNA. So there's no way we can just stretch out all of the information by in a serial way, right? So if you do the 100 base pair into your the 3 billion base pair reference genome, if you want to align like a first position, second position, and third position from 1 to 3 billion base pair position, it never finished, right? So you can make much more efficient way to recapitulate all of these DNA sequence information into certain dictionary format. So in that case, we use the hash-based mapping, right? So so hash, you can think about more vanilla ways. So most students probably use Instagram, right? So if you look at Instagram, you have hashtag, right? So hashtag, what hashtag doing, right? So if you put hashtag ice cream, then you click the hashtag, then you can link to any images or any posts related to ice cream, right? Because that hashtag is working for that collections, right? So in this mapping situation, you can also use the hash information. Hash is one of the basic things you can think about in a computational field. So DNA alignment also use the hash information. Then you can build up all of this index information into the one. Then you can easily track down the information from this, right? So if you get the read information T, G, G, T, C, A, right? Then you can easily think about, read the first 3 characters, and it's from where? It's from the third position, right? And then the TCA is from 6th position, right? So you can easily think that it matched to 3 and 6. Okay, TGG is the 310 and TCA is 6. So you can easily think that, this TGG, TCA probably comes from 3 to 6, right? Because it's not gonna be 10 to 6 because this is not in order, right? So you can easily identify where these from, right? And the question is, what about, the sequencing errors or the variance in the positions, right? So the read information TGT, TCT, not the A, right? So this A changed to T because of error or mutation, right? So mutation here is the SNP, right? So you have to think about the hash match TGG 310, the same, and TCT is the question, right? So we don't know whether this TCT is from, right? So that case, you have to think about in a different ways, right? We are coming back to this issue in a later section. Okay, so another question for you is think about how many k-mers we have to use hash mapping. So k-mers means here we use 3 k-mers, right? So k means that the numbers, right? So mer is the sort of, you can think about the character, right? So k-mer is the, we use the 3 characters, so we say trimers, right? 3 dash and more, right? So the way we can try is we can use the 1 character, but it's never gonna finish, right? And you can use the 3, so that has to be ATGC is the 4 possibility times 3, right? So we have about 4 to 3 times possibility from the DNA sequence information. So all of DNA sequence information has to falling into these combinations, right? But if you can use much longer k-mers, right? So you can use the 10 characters, right? So that has the much more possibilities, right? So it has, take some computations too. So long k-mer is good because it gives you very unique information for the mapping. So if you use the longer and longer k-mer, it's definitely easy to mapping your reads to the reference genome, right? Because it gives you the unique combinations that you can easily track down the positions of the reads, right? But then problem is, if you use the longer k-mers, you have much more, much larger number of possibilities. So you use much longer dictionaries, so your computers take some time to look up those index during the process, right? So it's not really good for the speed, right? So it's always balancing between the speed and accuracy, error rate, and your computational framework. So people can use the 3 k-mers, 4 k-mers, or 10 k-mers, but it depends on the algorithm and it depends on the situation. Okay, so that's one message. And the mapping algorithm. So back from this question. So how we actually do the mapping, how we actually do the alignments in sequence read. So here, mapping and alignments is the equivalent terminology, right? So mapping quality, in other words, alignment quality is the probability of how, accurately sequence reads map to the genome, reference genome, right? So mapping quality also use the Fred scales, right? So this is the, 1 in 100 or 1 in 10,000 or something. So it's also used the same Fred scales as the Fastq quality score. So you now you have the 2 quality scores in your same information, right? Okay, and this mapping quality is differed by algorithm, differed by softwares, and differed by experiments as well. So here we use the 2 experiments, right? And we use the 2 different algorithms, right? And the first experiment used the algorithm called bowtie, and the second algorithm called BWA, right? And the first experiment is based on the single N, and the second experiment is based on the paired N. And we can see the distribution of the mapping quality from the experiment, from the library, and from the algorithms as well. And if you are, if you're working on bioinformatics, or if you're working on genomics, or if you're working in the clinical, patient data set of the mutation or diagnostic field, you have to be familiar with, how accurate or how good your mapping quality from your patient sequencing data set. And you can always use the, the visual exploration and use, put it into the, the exploring the distributions as well. And how we do that? You come to the next semester class, learning R and Python. Okay, and then another problem is we can also think about the exact mapping for your alignment, right? So here is one example, is like a card and heart, right? Only difference is in C and H, right? In this case, we can say C substitute for H, right? So this is the only one distance differences, right? But sometimes the differences can be by deletion, right? So short and short, and this is the one differences, right? So if you put it into the DNA sequence, then this is the, let's say this is the reference genome, and this is your read sequence from the machine, then you can compare these two, right? So this can be mapping this way or this way, right? So it mapping to like here or mapping to like here. So you have to decide, you know, which way is the more accurate mapping for the reference genome. So how we judge that situation, right? So this case we do the distance method, right? And we do the scoring system for the matching, mismatch, and gap, right? So back to here. So this is the matching, right? So G and G is matching, and here is the mismatch, right? So reference genome doesn't have that, but this situation has it, right? So the read sequence has it, right? And also, and here is the gap too, right? So that has the T information, but here is no, right? So you have to come up with all of this scenario, then you can think about the scores for the mapping, right? So we put this reference genome sequence onto this part, right? TG, TTAC, GG, right? And then we put the read sequence information onto here, right? Then we can start from here. Then we can judge whether that is mapping or not, right? So we first initialized our matrix with a minimum scores of 0. So we all put the 0 at the first position. Then we're going to the second, we're going to the second, the first position. In that case, we are, T and G is the mismatch, but the rule is that we're not going to go below the 0, right? So we're not going to give any, the minus scores into this calculation. So we can just put the 0, right? And then once it's matching, so G and G is matching, then we put the match scores, so +3 here, right? And the next one is we can keep that calculation. So G and T, so this is not mismatch, but we can expect the matching at the next time. So we put the gap -2, and that's sort of the same situation. We calculate the scores out of this matrix. Then if you look at here and T, so if you go to A here, then this is the sort of gap, right? And here is also gap as well. And if you go to A and G, and if you take this, that will be mismatch, right? So you can put -3. So yeah, it's going to be 6, right? Then you can fill out all of this calculation into the end. Then once it's done, then you pick up the max scores out of this matrix. So that has to be 13, right? Then what you do is you do the backtracking, you do the traceback, right? So traceback, the rule is you trace back the next highest scores, right? So 13 is the highest. And if you think about the backtracking, you have the 3 possibility, 8, 10, 8, right? So the highest one is the 10, right? So you go 10, then 7, right? Then you go that way, 7, 9, 6, 3, and 0, right? Then you can build up the matching information to the reference genome. So from here, GTTAC, right? GTT, gap, and AC, and your sequence read is GTTGAC, right? GTTGAC. Then you can guess that your sequence rate is mapping in this way in a reference genome, right? So that is the judgment whether you go to this or go to this, right? So this is the traceback way you can do the alignment, right? So this is just a local alignment. So one part of your genome. So it takes, you know, the genome-wide situation in your computer, right? So if you do the all of your genome information, it will do the much quicker way, and also do use much computational research to finalize this mapping, right? So in early days, in you know 60s and 70s and 80s, and from the time people you know thought about how we do the exact alignment for the you know the sequence information. So I mean at the time people came up with the biochemistry development, molecular biology development. People see the future, right? So people talked about how we compare the similarity between the protein sequence and DNA sequence, right? So in that case, people use the algorithm developed for that purpose, right? So that time, I mean, the two famous algorithm. We're not going to cover very detail in this lecture, but if you are super interested in that, then you can starting from the comparison between the global alignment and local alignment, right? So global alignment is Needleman-Bunch algorithm from the 1609, and the local alignment is the Smith-Waterman algorithm, right? And this is largely used in the whatever biological sequence information in our field, right? So local alignment is specifically refining the local sequence, so very tiny portion of the genomic area, but global alignment is looking at more, introducing more gaps and introducing more mismatches and put it into more global ways. So of course, global and local alignment takes different, you know, the processing time and also different strategy allowing the mismatch and gaps as well, right? So it depends on the purpose, right? So if you want to complete the entire picture, then you go global. But if you look at specific position, then you go local. But also there is a hybrid concept. So you do the global alignment first, then you specifically looking at some portion with the local alignment. So this is sort of the starting point for the alignment algorithm for the sequence data set. But more high level, you know, the software. So high level means that you're not going to see the backbones. You're not going to see the, you know, the details of the alignments. We use very, you know, the superficial layers of the software. That case we call the high level, right? So if you think about the Google Drive or Dropbox, this is the cloud system for the high level situation. You're not going to see the computational background out of that because you're paying the money and you just use the very simplified interface, right? So that situation, same for the sequence alignment. So these days, if you do the, you know, the genomic research, you're not going to go into detail for the alignment method. So you basically use the software for that. But my case, I mean, if you are really super interested in the bioinformatics, you have to go to sort of details of the algorithm for your learning. Otherwise, you're never gonna get to the point, right? So, I mean, unfortunately, you know, our class is just for the one semester for the genetics, covering genetics and genomics. So we're not gonna cover very details of the genomics part. But you know, I mean, if you are working a very deep levels of the bioinformatics, then you have to, you know, go to the library and dig some, you know, text for the, you know, the sequence alignment. And there are some, you know, the, you know, third year or fourth year books for that purpose, right? But anyway, back to the alignment softwares. So there are several lines of that. So most, you know, famous one is BWA-MEM, and this is the developed by Hung-Lee, and one of the greatest algorithm developer. And also another algorithm like a Bowtie, Nova Align, Topaz, Tar, blah, blah, blah. And this is again all open source, and we can see their codes and also the different working application and approaches as well. So if you are super interested in those things, then you can follow up the specification of the things, right? Okay, so send format, I think we have to come back from here. I really should finish this one in today, but yeah, I mean, this is a bit sad. Okay, so we can resume from this part next Monday, or I can upload the video for your learning on Wednesday, but we don't have the class on Wednesday because it's a holiday. So I mean, yeah, so we're not gonna have the June class, but yeah, let me think about this part, and this is the another critical things for the entire part. So we can resume from here in replacement lectures, or the videos, or next Monday. I'll announce on the Slack. And hope you enjoyed the first alien, the sounds, computational things for the genomics, but I mean, this is not, inevitable things in your future. So please be familiar with all of this workflow. And also, if you have any question, leave yours onto the Slack channel, and hope you enjoy the rest of the week. See you next time.